everyone, welcome back to Biophilia, your all-time study mate. Today we are going to continue with the chapter Strategies for Enhancement in Food Production with its part 3. If you like our videos and want us to help out to reach out many more, then don't forget to like, share and subscribe to our amazing channel Biophilia. So continuing with the chapter, यहाँ पे हम पढ़ने वाले हैं about single cell proteins. अब single cell proteins क्या होते हैं? So generally when the people are going to eat meat, so जो non-vegetarians होते हैं, they are going to get their daily protein amount from the meat that they consume. So उन लोगों को प्रॉब्लम नहीं होती है, बट दोज पीपल हु आर वेजिटेरियन जो मीट नहीं खाते हैं उन लोगों को प्रोटीन डेफिशिएंसी होने के बहुत ज़्यादा चांसेस होते हैं इफ़ दे डोंट टेक राइट अमाउंट ऑफ प्रोटीन सोर्सेज. सो उन लोगों के लिए दोज हु हैव स्टार्टेड टू शिफ्ट टू ग्रेन मील, यानी जो वेजिटेरियन मील खाना शुरू करने वाले हैं, for them to consume the amount of protein they need, they are going to need about 3 to 10 kgs of grain. So 3 to 10 kgs of grains khana is not possible. So whereas if the person is eating meat, he can get it from 1 kg meat. So 1 kg meat can be sufficed in 3 to 4 days, or say if it is regular eater. But 3 to 10 kgs grain एक इंसान को खाना just for the protein source is not possible. So for that reason, those who people are eating or based on grain diet, they are going to be suffering for hunger and malnutrition. Hunger in sense that they are going to lack in protein sources. और ये तकलीफ is not like in smaller quantities. Almost more than 25% of human population is going to suffer from this type of nutrition hunger. So for that reason, जो लोग plant diet खाते हैं उन लोगों के लिए, those who cannot have animal proteins, for them the new source was found out that was called as single cell protein. So single cell proteins खाने की वजह से your daily protein content can be sufficed through this source. So let's understand about what it is, the single cell protein. So single cell protein जो है, it is going to be a protein rich biomass and which is going to be used as food, or it can be also fed to other animals as well. So not only for humans, it can be also given to different animals for their protein sources. So single cell proteins जो होते हैं, जैसे नाम है single cell, so बहुत लोगों को लगता है that single cell protein is obtained from organisms which are only single celled, but ऐसी बात नहीं है. Single unicellular organisms के साथ साथ multicellular microorganisms are also going to contribute for the production for single cell protein. So SCP, that is single cell protein, is going to be a good quality protein source and it is going to be poor in fats. So fats नहीं होने की वजह से if you are going to consume that much amount of protein, it is not going to affect your fat content in your body, but proteins are going to be enhanced. So as the diet or this single cell protein is going to be high in protein and low in fat, so that is the desired meal that the human should have. So features of human meal क्या होते हैं? Proteins ज़्यादा, that is the body should grow, the muscle mass should grow and fat should be less so that you do not become obese. So that is the reason for why the single cell protein has been a new fashion or new demand for the people, those who are going to having a healthy grain meal. So generally people are going to obtain this single cell protein by removing the nucleic acids, यानी जैसे microorganisms होते हैं. So if you consider for the bacteria, so bacteria is going to have the DNA or RNA material in it, और वो जो न्यूक्लिक एसिड मटेरियल होगा वो न्यूक्लिक एसिड मटेरियल को निकाल दिया जाएगा. फिर even if they are having any metals or any toxins in their biomass, all those will also be removed and only the protein part of that microorganism is going to be obtained, and that is nothing but going to be called as single cell protein. So single cell proteins के क्या क्या sources हो सकते हैं? So sources can be unicellular microbes as well as multicellular microbes. So first over here we are going to see the single cell protein sources that are fungi. So fungi में you can use yeast, that is Candida utilis, for obtaining the protein sources. You can also use mushrooms, and mushrooms are largely eaten by people. So large scale mushroom growing होता है. Many industries are going to be based on only mushroom growing. So mushrooms तो बहुत लोगों को पसंद है. There are many varieties of mushrooms that the people love to eat. So it can be yeast, it can be fusarium, it can be mushrooms, which are going to be the fungal sources for the protein. Apart from fungi, even cyanobacteria can be a source of single cell proteins. So cyanobacteria is like the example of a blue-green algae that is called as spirulina. तो spirulina is going to be a cyanobacteria जो बहुत easily grow हो जाता है, because it doesn't need any specific type of medium to grow into. It can also grow in like waste water में grow हो सकता है. So waste water तो you have it all around. So it can grow in wastewater. It can also grow in potato processing plants जहाँ पे potato process होता है. So like the companies which are going to produce potato chips, तो potato chips companies में जो starch वाला पानी होता है, for that starchy water can also be the medium where the spirulina can be grown. So many small water materials can be used. Even it can grow on straw, that is dried grass. It can be grown on molasses, यानी sugar factories के जो बाई प्रोडक्ट्स होते हैं उस पर वो ग्रो हो सकता है. It can also be grown onto animal manure, even in sewage. So इसके जो growth के जो areas है, these are not going to be very special areas. So they can be easily grown, and these are going to be very good source of proteins. For that reason these are specially been cultured, उनको special grow किया जाता है into big laboratories maybe, or there can be factories set up for the sourcing of the protein through the cyanobacteria. So these are now going to be these are going to be grown into such mediums, और उनको large quantities में protein rich food के जैसे उनको serve किया जाता है. So it can be protein rich, it can have minerals, it can have carbohydrates, vitamins, बहुत सारी चीज़ें हैं that the spirulina, that is a cyanobacteria, can provide. So these can be available in green powder format as you can see in the photograph, or they can be compressed and form into tablets. So there can be various sources through which you can obtain your protein intake. Third important source can be, that is bacteria. Very important bacteria जहाँ से बहुत majority में protein source किया जाता है, and that bacteria is going to be Methylophilus methylotrophus. This name is very important because this bacteria is going to have the ability to produce at high rate. उनका जो reproductive rate है या उनका biomass production rate है, that is going to be very high. So in no time these are going to be grown, and the produce that they form, यानी जो amount of growth है उसके comparison में जो protein sourcing मिलती है, that is going to be much more, and that is for about 25 tons of protein is going to be available from this bacterial species, that is Methylophilus methylotrophus. So for that reason even bacteria like these species can be used for sourcing single cell protein. Now the second topic of our study after single cell protein, that is going to be about tissue culture. अब tissue culture क्या होता है? Tissue culture is going to be the plant tissues. So as we are focusing majorly on the strategies for improving the plant materials, so over here the tissue culture of the plant majorly is going to be for the maintenance and for the growth of the plant cells. So ये जो plant tissue culture है, that is going to be based on the medium which is in vitro. In vitro है यानी laboratory conditions में also suitable conditions are provided, and the tissue or the cells are going to be grown up till they are capable of growing individually in the soil. So ऐसे जो cultures होते हैं जहाँ से the entire plant is going to be grown in vitro conditions, that plant part is going to be called as explant. So explant can be any part of the plant which will be taken out, उसको test tube में grow किया जाएगा, and that will be having sterile conditions. and it will be also having special nutrient media, यानी उनको जो भी चाहिए to grow. For example, they might need some hormones, they might need some energy sourcing. All that is going to be provided into the in vitro conditions. लैब में जो न्यूट्रिएंट मीडियम होगा उसमें ये एक्स प्लांट को रखा जाएगा, उसको ग्रो किया जाएगा, and finally the capacity to entire growth of the plant from that little cells that were taken out as explant. So explant से entire plant grow होने तक की capacity को बोलते हैं, that is called as totipotency. So totipotency, that is the capacity to generate whole plant from any cell, is totipotency. So generally tissue culture plants में follow किया जाता है, and it is not seen in the case of animals because plants के जो cell होते हैं these are going to be having more capacity to generate the whole plant. But if you try to go and test this totipotency in the case of animals, तो animals का कोई भी cell part निकाल के if you try to go and form the entirely new organism, that is not going to occur because the animals are going to be much more advanced. उनके जो differentiation process होते हैं these are going to be very highly advanced. So for that reason, if you try to take some of your skin cells or tissue from your hand, leg or whatever, so humans बनाने की कोशिश करोगे ऐसे, so it is not going to occur since plants are less differentiated or these are less advanced in the case of differentiation. Then for that reason totipotency, that is generation of whole plant, can be possible. So tissue culture तो हमने meaning पता कर लिया. Now let's understand some other points that we are going to see for this topic. So tissue culture में what are the environmental conditions that are required so that the success of formation of new plants from that little explant can be possible. So first of all, major important that will be the nutrient medium. So वो कहाँ पे grow होने वाला है, that is going to be important. So providing nutrition is going to be one of the major things to be remembered. So this nutrition is going to be provided from the culture medium. So culture medium में बहुत सारी चीज़ें ऐड की जा सकती है. That is carbon sourcing can be provided by the help of some sugar content. So sucrose can be added into it. There can be inorganic salts जो generally soil में available होती है. But since you are making your own little culture medium, so you have to add those naturally occurring inorganic salts. उनको vitamins देना ज़रूरी है, उन्हें amino acids provide करना ज़रूरी है, and also there might be some hormones which are going to be provided externally for the growth regulation. So it can be 2,4-D, which is going to be a type of auxin, and auxin we'll understand later auxin का क्या काम होता है, and the cytokinins, which is going to be another plant hormone, as you know from class 11. So these both are going to be provided into the nutrient medium. और जो nutrient medium होता है उसको acidic conditions में रखा जाता है, that is pH is going to be around 5.7. So if you are undergoing a tissue culture for plants, it is going to be acidic nutrient medium, but if you go for animal tissue culture for by any chance, then it is going to be basic in condition. So major difference between the tissue culture for both is going to be that the plant जो nutrient medium होता है that is going to be acidic 5.7 pH, but for the case of animals it will be basic in nature. So second environmental condition that is needed after nutrient medium, that is going to be sterilization. अब आपने उसको खाना पीना सब अच्छे से दिया है, but now it is important to keep the microbes away from that explant. So microbes को दूर रखना है, infection को दूर रखना है. So for that reason sterilization is going to be important. So sterilization किसका करना है? Everything that will be used in tissue culture. So what is going to be used? Everything starting from the explant जो grow होने वाला है उसको तो खास ध्यान रखना है कि उसमें कोई infection ना हो. So उनमें infection नहीं होने के लिए आप उसको grow करने के लिए जो भी चीज़ें use करोगे everything needs to be sterilized. So the culture vessels जहाँ पे वो grow होगा that needs to be sterilized. The nutrient media should be free from microbes, and also the instruments that would be using for the different purposes. Everything should be kept free from microbes. So अब कौन से टेक्निक्स से आप उसे स्टेरिलाइज करोगे? So the explant, so the plant that is being taken out, so that is going to be treated by antimicrobial chemicals, and since it is antimicrobial chemical, the process will be called as surface sterilization. So surface sterilization is going to be the process by which the explants are going to be sterilized. But अब जो बाकी की चीज़ें हैं, for example there are going to be vessels, the media and the instruments, all those are also going to be sterilized, but उनके लिए you are not going to use antimicrobial chemicals. Instead of that you would be using the steam, यानी इतने गर्म में आप उसको करोगे so that the microbes get inactive or they die. You can also use the dry heat, or you can use alcohol, and you can also filter so that they are becoming free from microbes. So the process that you are using here, that is steam use कर रहे हो, alcohol use कर रहे हो, all that is going to be called as complete sterilization. So vessels, medium या instruments जो नॉन लिविंग है, so नॉन लिविंग के लिए you are going to use complete sterilization. If you are going for the living source, that is explant, उसके लिए आप surface sterilization करोगे with the help of antimicrobial chemicals. So here you can see in the diagram about the steps that are followed for surface sterilization. So you can refer to that so that it will be easier for you to understand about surface sterilization. And here are two images which can be showing you about the autoclave. So first जो है वो autoclave के बारे में है, wherein you are going to use steam so that it becomes microbe free, and the next one is going to be the dry heat machine. So dry heat में you must have observed this type of machine is also used at the dentist's place. So dentist के जो instrument होंगे, all those are also going to be heat dried because of this machine. So this machine is also used at dentist place to keep the instruments microbe free or for sterilization process. The third important environmental conditions that need to be maintained is going to be aeration of the tissue. आपने उसको खाना दे दिया in the source of various hormones, in the carbon source you provided minerals, vitamins, everything. You also sterilized, kept it free from infection. But अगर आपने उसको gaseous exchange provide नहीं किया, then the explant is going to die itself. So for that reason, aeration of tissue is also going to be a very important step that needs to be provided. So if the proper aeration, yani proper amount of oxygen is provided into it, then the water that or the nutrient medium में अंदर continuous stirring होता रहेगा, and if it gets mixed, so in that conditions automatically the oxygen is going to be incorporated in the nutrient medium, and in that case proper aeration can be maintained into the cultured tissue. So with that the explant can grow with the help of the oxygen or the proper aeration. So over here the third point that you need to understand, so हमने देखा पहले कि टिश्यू कल्चर क्या होता है, सेकेंड हमने देखा कि what are the environmental conditions. Now the third part we are looking, that is how the tissue culture actually performs. So what methods can you use? So first method you can use, that is going to be the type of in vitro growth. So based on the type of in vitro growth you can use, that is Callus and suspension cultures. So हम उसको एक एक बार करके डिटेल में पढ़ेंगे, but for now you can remember that for the type of in vitro growth you can remember it can be Callus or the form of suspension cultures. On the type of explant that you will be using, यानी जो सेल या टिश्यू का मास आप कलेक्ट करोगे, so based on that collection from the location you can determine what sort of tissue culture it is. So if you are going to take the explant from the meristem, so that will be called as meristem culture. If you are using protoplast, then it is protoplast culture and the embryo culture. So we will see all them in detail one by one. So first is going to be about the callus and the suspension culture. So callus culture जो होता है, that is going to be the process in which the cell division of the explant is going to form a unorganized mass. यानी आपने समझो पहले कुछ cells लाए थे of the explant from the parent tissue, उसके अंदर cell division हुआ and it formed a irregular mass of cells. अब ये mass of cells जो होगा it will grow into size and this mass is going to enlarge and this enlargement is going to be seen on the agar plate. यानी ये agar जो होगा it is going to be the nutrient medium. So nutrient medium पे ये unorganized mass available होगा and that unorganized mass is going to be called as callus. So callus is going to be the unorganized mass, whereas this little plant that you had brought is going to be called as explant. So explant से growth होगे, cell division होके जो unorganized mass of cells बनेगा maintained on the agar, that is nutrient medium, is going to be called as callus. So that is nothing but callus culture. अब ये callus जो होगा वो further grow होगा and it is going to form the new plant. So that way the callus is going to be cultured. Now second जो method होता है that is going to be called as suspension culture. So suspension culture में क्या होता है that you are going to use a beaker or you are going to use some container wherein you are going to have the nutrient medium in liquid source. So liquid source में ये nutrient medium होगा, उसके अंदर आप explant को छोड़ दोगे and once you leave that explant into the nutrient medium which is liquid in state, वो suspend करेगा and since it is going to suspend, the culture will be called as suspension culture. So continuously उसमें aeration होते रहेगा so that the tissue does not die off. So continuous aeration उसको agitate किया जाएगा. Agitate यानी the air is going to be mixed continuously into the liquid medium. So agitation होगा with the mechanical force that will be having speed of about 100 to 250 rpm, that is rotations per minute. और उसमें different hormones add किए जाएंगे. For example, auxins can be added, 2,4-D can be added so that the suspension culture can be grown into the new plant. So as the tissue dries off and the dry matter is increasing, the nutrient material decreases. So this is a very important line, that is dry matter increases and the level of nutrient decreases. यानी जैसे growth होगी cell mass बढ़ेगा but the nutrient in the material is going to be decreased क्योंकि उसको consume करके ही grow करेगा. So for that reason, as the nutrient is going to decrease, it might happen that if you don't change the nutrient medium regularly, then the material will not get sufficient nutrient. हो सकता है कि वो plant पूरा grow नहीं कर पाएगा. So for that reason, to prevent the damage of the newly formed cells, the particular explant और explant के बाद के जो stages है उसको frequently new culture vessels में डालना ज़रूरी है and that process when you are going to change the particular nutrient medium by introducing it to new culture vessels is going to be called as subculturing. So subculturing is nothing but transferring it into the new culture vessels and that is subculturing. So पहले वो शायद हो सकता है कि छोटे से बीकर में हो, then you transfer it to a bigger medium because now the size has been grown up. वैसे करते करते that is nothing but subculturing. Now afterwards when you are continuously changing the culture medium, अभी आप उसको ऐसे stimulate करोगे so that the roots and the shoots are going to be generated. So root और shoots आपको artificially generate करवाने हैं with the help of controlled auxin and cytokinin balance. So it is important to understand that it is going to be controlled mechanism. Controlled है because whenever there is excess of auxins, the auxins are going to promote root formation. Whereas if you go for cytokinin, excess cytokinin will promote shoot formation. So auxins अगर आप एक टाइम ज़्यादा डालोगे तो root बनेगा. So root has been formed now. And once you add cytokinin, now the shoot will be formed. हो सकता है अगर आपने cytokinin कम डाल दिया, auxin ज़्यादा हो गया, if it is uncontrolled, then the root will start to grow but the shoot will remain shorter. So that is not going to occur और ऐसा plant grow नहीं करेगा. So for that reason it is very important that it is a controlled mechanism for the auxin and cytokinin balance. Now as soon as the root and the shoots are formed, they will be small plant like, और वो small plants जो होते हैं आप उसे क्या बोलोगे, it will be called as plantlets. So now these plantlets are going to be removed from the culture vessels, और अभी उनको culture vessels या in vitro conditions से आप उसे soil में जाके test करोगे. So once these are going to be tested onto the open grounds or open fields, that process is going to be called as hardening. So यहाँ पे हमने दो processes की बात की. First process that we saw, that was subculturing, जहाँ पे आप बार बार अलग अलग nutrient medium से उसे pass करवाते हो. And second, that is going to be hardening, wherein you are going to introduce the plantlets into the open fields. So once the hardening is going to be completed, आप उस टाइम पे क्या करोगे, उसे reduced light में रखोगे और high humidity पे रखोगे. So the conditions that you are going to change in the process of hardening, that is reduced lighting and high humidity. So ये conditions जब आप introduce कराओगे to the plantlets, they become much more stronger and adaptable to the harsh conditions that are going to be outside onto the open grounds. So before they are going to be taken out of the laboratories, उनको ये test pass करना होता है that they can survive into reduced lights and as well as high humidity, and once they are going to be capable for tolerating such conditions, now they are ready to go and fight against the natural environmental conditions outside the culture vessels. और फिर जब ये hardening process खत्म हो जाएगी, these plantlets are becoming much more taller and much more stronger. So ये जो plantlets होंगे अब वो उसे market में भेजेंगे so that people can grow according to their fields. So likewise tissue culture से और the main process, that is Callus or suspension culture से, these plantlets are going to be available into the nurseries or gardens. Now the second type through which you can use the tissue culture, that is meristem culture. मेरिस्टेम कल्चर जो होगा उसका जो एक्सप्लांट होगा वो एक्सप्लांट कहाँ से आएगा, इट इज़ गोइंग टू कम फ्रॉम द मेरिस्टेम. नाउ व्हाई आर यू गोइंग टू यूज द एक्सप्लांट फ्रॉम द मेरिस्टेम, बिकॉज व्हेन द कंडीशंस ऐसी आती है कि बहुत ज़्यादा डिसीज़ फैल चुका है, पूरे के पूरे फील्ड में डिसीज़ है, and now there is no time to re-sow the plants and to wait for them to grow naturally. So उस टाइम पे ये डिपेंड करना ऑन द नेचुरल वेज इज़ नॉट पॉसिबल. सो इंडिया में एक बार ऐसी कंडीशन आई थी वेर इन द एंटायर फील्ड्स ऑफ इंडिया वॉज इन्फेक्टेड बिकॉज ऑफ द डिसीज, and during that time it was not possible to wait and grow it into the next season, क्योंकि अगर ऐसे कर देते तो वो पर्टिकुलर ईयर में फूड फेमाइन आ जाती थी, फूड की शॉर्टेज हो जाती थी, that would have been a very disastrous condition. So during that condition this method, that was meristem culture, was introduced, जहाँ पे pathogen free clones was obtained through the meristem culture. Now why meristem cultures are going to be pathogen free? Because the meristem is going to be a region which is free of viruses. Free of viruses की वजह से दो है. The first, it is going to be in high concentrations of auxins. So auxins बहुत ज़्यादा होते हैं into the meristem region. And second, meristem regions are going to be the places जहाँ पे rapid growth होती है. So rapid growth, or these are the young areas जहाँ पे cell division बहुत ज़्यादा है. So since there is rapid cell division, there is no chance that the viruses can go and attack into each and every cell of that meristem. So meristem is most of the times free of pathogens, free of viruses. और अगर आपने ये मेरिस्टेम को हार्वेस्ट कर लिया है, मेजरली द हार्वेस्टिंग इज डन फ्रॉम द अपाइकल मेरिस्टेम. Apical meristem is going to be the tip of the shoot. So tip of the shoot, say when the explant is collected along with 1 to 2 leaf primordia. Primordias are going to be the leaf buds which are going to be grown up into the new leaf some or the other time. So ऐसे अपाइकल मेरिस्टम और लीफ प्राइमोर्डिया को कलेक्ट करके स्टेरिलाइज करते हैं, and once that explant is now taken or cultivated, these are going to be cultured over the medium, nutrient medium में laboratories में उसको in vitro conditions में उगाया जाएगा. And since you are going to provide the nutrients all over the time, there is going to less time consumed to have full growth. So बहुत कम टाइम में फुल ग्रोथ हो जाएंगी. एंड ऑल्सो अंडर द असेप्टिक कंडीशंस यानी पैथोजन फ्री कंडीशंस मेंटेन किए जाएंगे with the help of sterilization. So from that reasons you can have that particular crop into that particular time phrase. अगर आपने ये मेरिस्टेम कल्चर नहीं किया होता, what could have happened? It could have happened that you might have to wait for the next season so that you can sow into the soil, and again you wait for it to grow, और उस टाइम तक famine आके most of the people would have died. So instead of having those situations, because of advancement in science, you can have meristem culture, and within no time you can have new plantlets which have been already grown simultaneously, and now you can protect the particular humans for undergoing these famine conditions. So generally meristem cultures जो होते हैं वो बनाना के लिए, शुगर केन के लिए या पोटैटो के लिए यूज़ किए जाते हैं. So the small plantlets of bananas are going to be produced into laboratories. The plantlets will be provided to the nurseries, और nurseries में से जब आप उसे खरीदोगे आप छोटे छोटे plantlets को sow करोगे instead of sowing the seeds. So if you can see up here in the photograph, what is going to happen, you are going to take the little part of the shoot टिप को आप न्यूट्रिएंट मीडियम से स्टेरिलाइज करोगे, डिस्टिल्ड वाटर डालोगे, एंड देन यू आर गोइंग टू incubate under the fluorescent light. आप उसे grow होने दोगे, there will be root development, and from the root development the shoot is already been developing. They will be transferred into the pots. So pots में आपने क्या डाला, pots में आपने छोटे छोटे plantlets डाले. आपने seed डाला नहीं, you did not wait till the seed to grow into small plantlet. You already sow the little plantlet into the pots, जिसकी वजह से time बचता है. And this meristem culture can be producing large amounts of these little plantlets. So this can be a beneficial step when there is a disease outbreak. So disease outbreak के time पे meristem culture can be a very big advantage for producing the different types of plants. Third important type is going to be protoplast fusion, also called as somatic hybridization. So protoplast fusion, जहाँ से नाम से पता चलता है, that is protoplasts of two cells are going to be fused. So how is it going to be done? So protoplast क्या होता है? First of all you need to understand that. So when the plants are going to have their cells, वो plants के cell पे cell wall होती है. So when the cell is going to be removing its cell wall, that is going to be called as protoplast. So it will be called as a naked cell. So that naked cell is nothing but the protoplast, which is going to be treated by the enzymes of pectinase. Pectinase or cellulase are the two enzymes which are going to remove the cell wall. So remove the cell wall, जो cell से cell wall निकल गई, तो now it is going to result into the formation of protoplasts. Protoplast यानी naked protoplast because the cell wall has been dissolved. And once the protoplast is obtained, they are going to be fused by two processes. The first process that can be used is electrofusion. Electrofusion is going to be the method जहाँ पे high frequency electric fields को pass किया जाता है through the medium so that these protoplasts come together and they fuse. Otherwise you can use chemofusion, and in chemofusion you can use sodium nitrate or you can use polyethyl glycol. So you can use PEG, that is polyethylene glycol, or you can use sodium nitrate. These are the two chemicals that you can use for the protoplast fusion. And electrofusion, that is high frequency alternating electric field currents. So because of these two reasons you can mix or you can fuse the protoplast. और जब two protoplast fuse हो जाएंगे तब क्या होगा, there will be a new cell formation or a new protoplast formation that is going to be called as a hybrid. protoplast. So hybrid protoplast जो होगा, it is going to be called as cybrid. What is it going to be called as? It will be called as cybrid. So since it is combination of two cytoplasms combined होंगे, that is the reason it is going to be called as cybrid. And once this is formed, the cybrid is going to be developing और वो new plant में convert हो जाएगा. So somatic hybrid formation में you can have two variants. The first variant that you can have is synkaryon. Synkaryon का क्या meaning होता है? Synkaryon means the nucleus are going to fuse. So if the nucleus are going to fuse, that means the two cytoplasms are completely fused. आपके nucleus दोनों के दोनों fuse होके एक हो गए. That is synkaryon. If it is heterokaryon, hetero means different and karyon is nucleus. So if there is two different nucleuses found in the fused gene, in that condition it will be called as heterokaryon. So for the first time जो somatic hybridization या first time somatic hybrid जो था वो किसने obtain किया था? It was obtained by Carlson. Carlson was the person who had first obtained the somatic hybrid in the Nicotina species. Nicotina क्या है? Nicotina is going to be the tobacco. So tobacco के दो species के अंदर somatic या protoplasm fusion करके he had obtained the first somatic hybrid. Now after the first somatic hybrid, many other experiments were conducted to mix the two characteristics through the protoplast fusion. So एक जो ट्राई किया गया था that was the intergeneric somatic hybrid. क्या होता है intergeneric somatic hybrid? If you try to break and understand the meaning, inter and generic, that is between the two genus. If it is happening between two different genus, then that was the experiment which was going to yield you a new type of plant that was called as pomato. So pomato जो है, pomato किसके बीच का hybrid था? It was going to be a hybrid between potato plant and it was a hybrid between tomato plant. So now you know potato और tomato दो अलग अलग genus से आते. So between the two genuses the hybrid was going to be formed, but ये pomato succeed नहीं कर पाया. There was no success for the formation of pomato because it was not having the desired qualities to be commercially utilized. वो laboratory conditions में या in vitro conditions में when everything was provided, all the favorable conditions were provided, उस condition में ये pomato continue कर पाया. But once it was placed for the hardening process, यानी आप उसको open fields में छोड़ दिया गया to grow alone on its own, उस टाइम पे ये pomato plant survive नहीं कर पाया. So for that reason now you don't have the pomato species या फिर pomato अगर आने वाला था. So scientists had thought about that the tomatoes and the potatoes, what characteristics can you mix in them? The potato is hard enough, उसका shell life ज़्यादा होता है. So potato does not get they do not shrink, they do not get, वो पिचक नहीं जाते हैं during transportation. वो tomatoes कैसे होते हैं? Tomatoes जो होते हैं these are going to get वो पिचक जाते हैं, वो टूट जाते हैं, वो जल्दी सड़ जाते हैं. So for that reason they were going to mix these two characteristics कि वो potato जैसे थोड़े hard हो जाए so that the transportation becomes easier and also the shelf life could increase. So for that reason they were forming the pomato. Also this intergeneric fusion was going to be considered for the hybrid between tiger and lion. So that was going to be called as liger. So liger जो एक था experiment, it was going to be the fusion between lion and a tiger. But again since the permission is not granted to have experiments on animals, for this reasons the liger was not going to be a successful experiment. But plants के ऊपर उन्होंने success किया था until it was in vitro. दो commercial utilization के लिए ये pomato succeed नहीं कर पाया. So that was about the protoplast fusion. Now let's understand कि ये सब जो हमने अभी पढ़ा इतने डिटेल में about everything regarding tissue culture. तो टिश्यू कल्चर के बारे में पढ़ के इनको रियल लाइफ एप्लीकेशन में कहाँ यूज़ करते हैं. सो फर्स्ट ऑफ ऑल यू आर गोइंग टू यूज़ देम फॉर द रैपिड कोलोनल प्रोपोगेशन. रैपिड कोलोनल मतलब जहाँ पे ऐसी कंडीशंस है वेर इन यू नीड टू हैव अ लॉट ऑफ propagation in very short durations. So large number of plants चाहिए in very short durations. वैसी conditions में tissue culture can be a very big advantage. So if you are going to ask for a method which is capable of producing thousands of plants in no time, in that conditions which are going to be identical to the parent plant, आपको एक ऐसा हमने as we have studied about in the previous videos of this chapter, जहाँ पे we studied about plant breeding. सो प्लांट ब्रीडिंग करने के बाद आपको एक ऐसी सुपर स्पीशीज मिली है जहाँ पे आपको जो भी कैरेक्टरिस्टिक्स चाहिए थे वो सब मिल गए आपको. नाउ एज द डिमांड हैज़ इंक्रीज फॉर दैट पर्टिकुलर स्पीशीज, नाउ यू वांट टू सेल इट एट अ वेरी लार्ज स्केल. सो लार्ज स्केल पे जेनेटिकली आइडेंटिकल प्लांट चाहिए, यानी ऐसा ही प्लांट चाहिए जो कैरेक्टरिस्टिक्स आपके बनाए हुए या आपके ब्रीडिंग किए हुए प्लांट में है, आपको वैसा ही प्लांट चाहिए. बट यू नीड इट इन लार्ज क्वांटिटीज एंड द डिमांड इज इंक्रीजिंग, सो टाइम नहीं है ज़्यादा, so you cannot wait for natural propagation. So in that conditions you can have this tissue culture used so that there is a rapid colonal propagation. So it will be producing large amount of plants in short duration and also they will be genetically identical. So ऐसी conditions में जो plants बनाए जाते हैं आप उसे क्या बोलोगे? You will be calling them as soma clones. So soma clones are going to be formed at thousands lakhs of numbers, और ये सोमा क्लोन्स जो है वो ऐसे बहुत सारे प्लांट्स में ऑलरेडी बनाए जाते हैं. दैट इज इन द केस ऑफ टोमेटो, इन बनाना, इन एप्पल, एंड दिस आर गोइंग टू बी द एग्जाम्पल्स जहाँ पे योर tissue culture has been a very big success for the commercial scaling of these plants. Second important application that you can use, that is tissue culture can be used for having pathogen free plants. So जैसे हमने पढ़ा था about meristem culture. Meristem culture में the meristem is the region of the plant which is free of viruses. So since it is free from viruses, it can be free from pathogens as well. So जहाँ पे disease outbreak हो चुका है उस conditions में diseased plants के meristem culture किया गया, and with that the recovery of the healthy plants can be possible. So diseased plants से healthy plants grow करना is going to be possible because of the tissue culture. So one of the examples जहाँ पे tissue culture use किया गया था for pathogen free plants, that was to have resistance from the tongro virus. Tongro virus जो spread होता था from the leaf hopper. So it was the vector that was going to be transporting the particular tongro virus. और टोंग्रो वायरस जो राइस में होता था कहाँ पे अफेक्ट करता था? इट वाज अफेक्टिंग ऑन टू द राइस प्लांट, और उसकी वजह से बहुत सारा प्रोडक्शन लॉस हो जाता था. एंड फॉर दैट रीजन देयर वाज अ रेजिस्टेंस फाउंड फॉर टोंग्रो वायरस दैट वाज स्प्रेडेड बाय लीफ हॉपर, एंड दैट वाज पॉसिबल बिकॉज ऑफ द टिश्यू कल्चर. So now here we have completed the complete understanding about two major topics of this particular chapter. पहले हमने complete किया to understand about the single cell protein, wherein we had to understand कि सिर्फ vegetable या फिर grain sourcing से protein नहीं मिलता है. For that reasons people need to depend on some other external sources wherein the single cell protein can be advantageous. Second हमने पढ़ा about tissue culture, and now we have completed the applications for the tissue culture. So with this we complete the one of the major aspects of this chapter जहाँ पे हमने plants के बारे में पढ़ा. So if you like this video, अगर आपको इससे कोई मदद मिली है to understand the concepts much more better way, then don't forget to like, share and subscribe and stay connected to Biophilia so that we can study this chapter in one more aspect regarding animals. So हमने plant के related strategies बनाई so that food production can be improvised. Similarly for the animals we'll also continue with this chapter. Till the time we come back soon with the next part of this chapter. Keep studying, keep smiling. Thank you everyone. Bye bye.